The overall goal of this procedure is to visualize events of herpes virus transport and spread using fluorescent protein fusions in primary neuronal cultures. This is accomplished by first establishing sample incubation at physiologically relevant temperatures on a microscope capable of fast-switching fluorescent illumination. In the second step, the parameters of the fluorescence imaging are set up to limit any cellular photo damage while acquiring reasonable data. Next, either sequential acquisition of the fluorescent channels is performed, or multi-position, multi-image tiled frames are acquired. In the final step, the data is analyzed and reformatted for presentation and publication. Ultimately, this method of live cell imaging is used to track the movement of fluorescent viral particles within neurons and to follow infection as it spreads between cells.
This method can provide insight into the transport and spread of herpes virus in neurons. It can also be applied to other dynamic events in cell biology and microbiology, such as the endocytic uptake of particles or cell migration.
Before any imaging experiment, connect and begin warming the microscope stage-top incubator. After 30 minutes, turn on the microscope, the transmitted and epifluorescent light sources, and the automated hardware controls. Then, open NIS Elements, the microscope control software, to connect to the microscope. Here, a screenshot of a standard user interface in NIS Elements is displayed.